Well, welcome everyone. I think we're going to go ahead and get started. It's a little bit after 1. We want to be conscious of your time here. So thank you so much for showing up. This is our postdoctoral seed funding event where we get to hear the research that you have done over the time, and for the new cohort, what you're planning to do. So the way that it will go is we'll first hear from VPR Snyder, and then we will have the cohort from last year. You'll share your presentations that are already up here on the desktop. And then we'll take some questions. And if we need a break, we can have a quick break for some snacks that you'll see in the other room there. And then we'll hear from the newest cohort. And you will just come up. You don't have to have no PowerPoint slides, but you'll just share and describe your research. And then VPR Snyder will close it out. So I'll turn it over. Thank you.
Well, thank you all for being here. I'm Scott Snyder. I'm the Vice President for Research and Economic Development here at the University of North Dakota. I've been in my position now for about 3.5 months. So still very much figuring things out, and I'm absolutely delighted to hear what some of our best and brightest minds on campus have to say. And I'm very interested to hear what research projects you're having or you have going on, and what other thoughts you may have. I look back at my career and think of my postdoctoral years, which I did at the University of New Mexico, as really kind of one of the highlights of my career, because there's something about being in a postdoctoral position that's different than being a doctoral candidate, right? Is all of a sudden the research is totally your job, and you realize how interesting and fascinating, but sometimes frightening it all can be, because you want to make certain that the data is there for the publications, but you also learn at that time really. Was I saying something bad? No problem. No, you really, you learn, I would argue, how to become a professional. And it's, a postdoc sets you up really well for getting external funding, sets you up really well for understanding what a professional researcher does, wherever your next step takes you, whether it be into an academic job or into industry or elsewhere. These years are really formative and kind of continuing to develop you on your professional trajectory. So I thank the postdocs here for joining us and for taking this opportunity at UND, and I thank the faculty mentors for doing all the hard work that it takes to bring somebody on board and to kind of get them thinking in big and expansive ways. So thanks a bunch, and I look forward to hearing what you have to say.
All right, thanks. The order will, Paul, you'll come up first, and then Pantea, you'll be second. And then Xiaodong be third. Susan, and then Dmitri, or the postdoc, Dmitri's postdoc. So Paul, thank you.
Afternoon, everyone. I'm Paul Snyder. I'm part of the aviation program or aviation college at UND. And then I have Srijeth Nair in the back here as well. And he's part of the aviation and is the co-PI. So our project was quite a few years ago that we put in a postdoc for 2 years ago at least. As you can see, I mean, it's some pretty key words as far as the things that are going on in the UAS world right now, from machine learning to counter UAS to data fusion and of course swarms as well. And so they're all kind of pretty buzzwords that are things that are needed within this realm of aviation for sure. And of course, it crosses all the disciplines, which makes it even more exciting for the opportunity. We started out trying to get somebody, and I thought, we actually hired somebody April 1st of this year. It's Michael Ulrich. He's right in the back there. So he started, about a month ago right now. So there was a part of me that wanted to say, oh, we just got the person hired, and that's the last slide, and next person can go. But I thought that wouldn't be very thankful or grateful for the opportunity to have someone that we can focus on research for the next few years and to do good work. And so as we tried to get that postdoc, we went through a couple iterations of that. It was at $55,000. It was trying to get, somebody with some pretty good experience to be doing things related to swarm and to MLAI type stuff. It was counter UAS. And so there was a need for a lot of requirements to make that happen. And we really struggled with that through multiple iterations. And then we were requested the opportunity to reduce the requirements to enable us to get somebody that we could be able to start doing research and working on these things. And that was approved, and then we were able to hire somebody. So we're pretty excited to have Mike in the back working since April 1st. Srijith especially Srijith and I with him did not slow down in trying to do work, even though we hadn't gotten that person. We were just like piling up stuff for Mike to do when he got here. And so we kept kind of moving forward because we're somewhat passionate about some of these, opportunities and things that's going on. And we had the opportunity a couple years ago to get a small $10,000 grant from aerospace to start looking at counter UAS systems and acoustic, counter UAS systems. And so Srijith led up some work in that area, and it was particularly to counter UAS and using acoustic again to be able to hear other aircraft and basically enable us to track those aircraft, those unmanned aircraft, and then theoretically in the future would be able to use another UAS that could actually go after them, so to speak, and do the mitigation side of it. That's what we're kind of working towards. But what was so fun about this is that as we started doing these acoustic sensors, we started seeing opportunities in other places that weren't related to counter UAS. And we moved into an agricultural location where we started looking at, could we use these acoustic sensors to track bee movement? And by listening to the bees, know where the bees are at on a given field. And a couple of years before that, we had done some work with a different company kind of in this area. So we had the idea already out there, but using acoustic was not one of them. So the idea is we could be able to put across the field, we could put these acoustic sensors that would then track the sound of bees and identify where the bees are traveling to and where they're going from. And then we can identify where do we put the beehives so that we can have greater pollination for that field so we can get higher yield rates, for a particular crop. I think there's over like 100 different crops that need pollination. So knowing where to place those bees could be really beneficial. That opened up a door to some other insects that make strange and odd noises as well that we could use as well, but we haven't got there. We didn't get that one, but they were very excited about it. And they actually came back to us and said, hey, we think you should look at these areas and maybe kind of push into these different types of insects where we really could use some help on. And so we're still exploring that and trying to figure that out. But that was a first thing. And then Sridhar had some friends that we're able to work with over in India and develop another submission not too far after that as well. And knowing that we were actually hiring the position for Mike, and we were looking at some prospective people to come in to do it, with our FAA Center of Excellence, we were able to put in a proposal in. I was able to put time for him to be a part of that project. And that's coming up in October of 2024, in which we're looking at disaster response. And counter UAS is part of that, the disaster response, because if you have a fire and all of a sudden there's UASs flying in that area, that will prevent the bombers that have to come in to drop the water, to do whatever needs to be done to put that fire out. It stops all aviation activity and really can be a hindrance and a danger to the people on the ground. And so counter UAS systems can be one way to identify, who's in that area. And then possibly in the future, you might have to mitigate that if there's, a serious danger involved. And so that's a project that is under review, but we're pretty darn sure that we're going to get that. So we're excited for that project to move forward in the fall. So then he started April 1st, and we said, Mike, guess what? There's a seed grant next week. It's due on the 5th. So get to work. So in that week, he helped to develop a seed grant with Srijith's help to develop a digital twin test bed for UAV swarm for concept to flight. And so, there's so many things that we can't do. We can't do in real life, the jamming, the things like that. I mean, the laser mitigation things, but can we bring some of that, like the jamming, things like that? Can we bring that into this digital world and actually start testing this stuff out? And so we put that seed grant together. And then on April 15th, 2 weeks in, we found that we were awarded that from Aerospace Foundation for $10,000 to continue that work. So we definitely hit the ground running to have him start doing some of this work there. As far as articles submitted, 2 weeks, Mike, you got to get on that. I know you've had a month now actually, so no, but so we're not quite there yet since this position just started. Mentioned this already, we got that award. And so we're really looking forward to that. There's a couple aspects of it that we'll have him involved on as well. But the big one is what I've already mentioned. Already getting past my time. A couple things in addition to advancing research, or a summary to that initial developing that digital twin test bed for UAV swarm. We've also started to work on him advancing the research by developing, you call it a literature review, but really having Mike's, you already got the outline laid out. But what is, counter UAS? What is happening right now? What are all the things going on? And some of the best literature that's out there right now is like 2019. And so can we create a really good document that identifies what are the ways that we can identify and we can mitigate and all those kind of things with counter UAS? What's out there right now? But then taking it a step further and start developing what is the future technologies that we're going to have to fight against in 5 years? What's coming out as far as that goes? And then how we're going to counter those counters that are coming to fruition, right? And so if we can start developing that now, become more of an expert on those areas, then that can then be used for future research projects, can be used for knowing where to start focusing in on, opportunities as they arise and being that lead and understanding that. So we're going to create 2 documents, one for counter and then one for swarm as well. And so we'll see what that kind of comes out to. We have, like I said, a great vision of some really great documents, but the best ones were last updated in 2019. So there's really a need to kind of grow that out. And then as far as developing, support developing the counter UAS system, we use acoustic and vision information to identify and track intruders using ML. So we've already done this work and Surijith, I'm sure you could answer a lot more questions than I can, but we've been able to engage students, some undergraduate students as well, to help us to build that out. So we've done some things indoors with the idea of acoustic sensors, but we want to get to the point where we're doing outdoors, and same thing with swarms. We're getting to some points of doing some small things indoors, but we're hoping that we can get that to be outdoors eventually as well. And then developing some tools that will enable us to create a common operating system, so to speak, where all this data can be fused in so we can have all the data. So then theoretically, we could have cooperative aircraft that we control and uncooperative aircraft that we don't control, and be able to move the cooperative aircraft out of the way for a counter UAS type mechanism, like a drone, to go from one spot to another, for example. So a lot of fun things to think about in the process. Right now, we're working on 2 more proposals. Our goal is to try to put out a proposal a month, and that's what we're aiming at and trying to do. And so we got June and we got July taken care of, and we'll see what the rest of the summer brings. And with that, my quick rundown. If there's any questions for Surijith, he's here, or myself, I'd be happy to answer them afterwards or now.
Hi, everyone. This is Pantea Tavakolian from Biomedical Engineering program. The area of research I have is more about pressure injuries detection or prevention using mattresses. So there was a pivot in the research that we had. We submitted this for non-melanoma skin cancer detection, but usually when I want to take on a project, I go through regional ICOR and do customer discovery to see whether, to test my hypothesis and see whether there is a path for that device, that technology to be commercialized. And I didn't see this path for non-melanoma skin cancer, but I saw that for pressure injury detection. So that was the reason that we pivot towards that. We got this seed fund in beginning of 2022, but it took me also 9 months to hire my postdoc here. We advertised it twice, but 24 people for the first round out of 24, none could be hired. We advertised it, and then Rana applied for it, and we got Rana Shaban through that. She's going to talk to you about the work she helped me with. The past year, we applied for 6 grants. 3 were not successful. We're going to hear from 3 other this summer for 2 different line of research. She mainly helped us with pressure injury prevention assessment scanner, that she's going to talk to you about that.
Hi, everyone. I'm Rana, as Pantea just introduced. I'm a postdoctor at the Biomedical Engineering Department, and I'm happy to be here. I've also earned my master and my PhD from the electrical engineering department here at UND. So it's been a long journey here with UND, and I'm happy to be within this community. I'm here to present my work during this academic year, and this presentation is based on the following topics. Mainly, I was a co-PI with Pantea on the NIH grant for pressure injury early detection using spectroscopy scanner device. What is pressure injuries or bed sores? Bed sores are caused due to prolonged stay in hospitals, nursing homes, on the bony prominences in the body, like shoulders, heels, sacrum areas. Over 2.5 million US patients develop pressure injury annually. And sadly, 60 of those die. Also, there is lots of expenses for pressure injury in the US that surpasses $26 billion. Moreover, the individual treatment for pressure injury can cost from $21,000 to $152,000. And the pivotal factor to prevent pressure injuries is early detection. Pressure injuries and early detection is far more cost-effective than the treatment of pressure injury, because the delay between the occurrence of pressure injury and the visual detection of the skin discoloration of pressure injury hinders the nurse's intervention. What are the current methods already available? Visible skin tissue assessment, STAs, is the gold standard in the healthcare system. It's mainly visualize and evaluate skin discoloration, dryness, or any indicator or palpable indicator of injury. But there is a lot of limitations in this method, as it's hard to early detect stage 1 pressure injury in dark skin tone patients. Also, it is subjective. There's 2 devices to detect pressure injury. The SEM proviso is in market, and it can early detect pressure injury 5 days earlier than the visual assessment. Its accuracy is 80% because it's based on electrical impedance and there is noise in that. And it only detects the moisture and the water content, so it depends on one biomarker. There is an IR-MED device. Technology, it depends on the infrared, but it's also not currently in the US market. It's not yet approved in the US market. That's why we propose the PIPA, Pressure Injury Prevention and Assessment. It's an handheld device, multimodal spectroscopy scanner with an AI-based technology to objectively detect skin biomarkers for pressure injury, multiple biomarkers, not just one biomarker, using the visible light and the near infrared. And it will simply give the user a risk score, like a thermometer, and it will give you if it's risky, this area can develop pressure injury, and it's easy to use by non-expert nurse or any user in the healthcare system. PIPA mainly, it's a non-invasive and can qualify the changes in the skin biomarkers due to how transparent and thin the skin is. For a depth of 2 millimeters or less in the skin, the visible range between 400 and 800 nanometer can detect multiple biomarkers in the epidermis layer affected by pressure injury. And for the deeper inspection of the dermis and the subcutaneous layer, we can use the near infrared part, which is from 800 to 1700 nanometers, and it can detect the properties that are affected during the pressure injury development in the skin. PIPA can also evaluate the pigmentation, the melanin content of the skin at wavelengths 970 nanometer and 1450 nanometer. We can measure the water content and the moisture. Also, between 500 and 600 nanometers are good indicators for the oxy-deoxy hemoglobin content. And the fluorescent excitation at 365 and 405 are good for the collagen level assessment of the skin. So PIPA is really promising technology for detecting early the pressure injury, which will prevent hospitalization and save millions of lives and dollars. Currently, we're finalizing our IRB protocol with the experimental procedure for our clinical study that will take place hopefully during this summer when we get the approval. Also, there is a journal paper submission entitled as Diagnostic Technologies for Pressure Injury Detection, a systematic review for the Journal of Tissue Viability by Elsevier. That will be submitted within the next week. As Panta just talked about how the experience of joining NSF I-Corps and how the customer discovery is really helpful to hear from the users and for the customers, what is the gap in the market? What technology they're really looking for? We interviewed 19 wound care specialists and doctors. And our hypothesis at the beginning was that our device can help early detection of foot ulcer neuropathy. And we found that actually our device can cover the market gap in the pressure injury and foot ulcer instead of neuropathy independent of skin color, not the neuropathy. That's how it's really useful to go out of the lab and talk to customers, talk to whoever the doctors, wound care specialists, what do they need, what they are looking for. I also was fortunate when I first joined the BME lab in the fall. There was already technology. They were going for I-Corps and customer discovery. That was not for the detection of pressure injury. That was for the prevention using an air mattress, small cell air mattress for the operating room to prevent pressure injury in the US hospitals. And we discovered during our 20 interviews in those 4 weeks that not just the OR, the operating room, can be a market segment for this technology, but also the ICU can be another market segment. It was really fruitful experience. And here's the certificate for our team. As I talked before, our future work this summer is to go for the clinical study once we get the IRB approved. Also, we have plans to attend the National I-Corps NSF program, and we're gonna do 100 interviews, not just for the customers, and it will be for building our business canvas for the whole technology. Also, we are planning to apply for the NIH Phase 1 SBIR in September in 2024. Overall, it was a great experience between, I would say, preparing for proposal, writing grants, attending courses in the biomedical engineering department, which is innovation-based learning courses, and seeing how students are motivated to learn and work simultaneously with industry, and advising those students, whether undergraduate or graduate students, and see how this, I would say, since my graduate studies, it has been an evolution in the learning system, and see how students are not just studying in the lab, they're looking for technologies, they're looking for developing devices. It will help patients in the healthcare system. It will revolutionize the new healthcare system. I'm happy to be here, and I'm looking forward for the next academic year and hopefully get our grant accepted. Thank you.
Hopefully, and disclosure will be submitted, and maybe later on a patent will come out of it. But yeah, this is also a collaboration with the company as well.
Okay, good afternoon, everyone. My name is Xiaodong Hou from College of Engineer and Mines Research Institute. So my topic is coal-derived carbon materials for clean energy. Little bit different than the postal topic. I will explain why. A little bit introduce about our institute. Actually, our current name is IES, Institute for Energy Study, but we are in the process of reorganize, rename it, rebrand this. And as a team, we were when the last year the Excellent for Research. So you might heard about this, but we don't use that name probably very soon. Anyway, we are the one of the initiative under the College of Engineer Research Institute. So there are three key research areas. The very first one is the rare earths. You must have heard about this project. There is three pineal plant nearby the Red River. And then the second one is the carbon management and the fuel technology. This is our the core technology we have been working on, mostly dealing with coal combustion or carbon CO2 capture or just energy system. And the third one, which I'm mostly working on, is the battery technology, which I will explain the next nice. And this is what I'm mostly doing with my chemistry and materials background, trying to find a sweet spot between, carbon ore. We don't call this coal. We most use the carbon ore. They don't like this name. And between this and the clean energy, as you know, North Dakota energy and agriculture, these two economic partners. And with the carbon decarbonization, mission, a lot of pressure under the whole, value chain on this. So we are trying to see how we can help with this. About 7-8 years ago, we're thinking about how to develop, carbon materials from this, this huge resource. North Dakota is the largest lignite resource in the world. So mostly my research is about making carbon materials for rechargeable battery, lithium battery, or similar. And so, and also involved some other stuff like CO2 capture and hydrogen is something which originally I proposed for this postdoc. This topic is trying to make a catalyst to make a green hydrogen using the coal as a feedstock. So the carbon matrix, so we're going to come from the coal, and then the main selling point we see is the cost, and also the non-precious metal. We are proposed to use nickel rather than the palladium, which is currently state-of-art catalyst for making this green hydrogen. So MOF, that is a kind of very porous and high efficient matrix for the catalyst. So another reason I want doing this is currently under the chemical engineer department, we have this nice facility that has been set there for over 15, 20 years probably. That's under Dr. Michael Mann and by NSF award. And currently, is still functional, just needed some, replacement on those electronics. And so that's why I was trying to see if we can get a revive this nice called hydrogen and power lab. And the major advantage of this lab is this is not a very small device, not like a lot of single faculty were built in the lab. This is considered as quite a large size. So you can see the electrolyzer is 6 kilowatt. The whole day running this, you probably can generate a couple kilogram hydrogen, which is a pretty large size. And by the way, the very first PhD who built this is now a technical leader in the Unreal, who is doing the same thing. We actually contacted them last year, and he built his job is build probably a 30 times larger, the exactly same system in the Unreal. So I was so excited about this. Another PhD work on the same system is on the Nissan, are also doing the nitrogen. So that's a very good pioneer work on the hydrogen back to maybe 15 years. So, but we're getting a little bit challenging, probably like many other people trying to find a very good set of expertise on this. The major challenge is we probably can find a very nice candidate who can work on very small device and chemistry or engineer wise, but in terms of running the larger system, there's very few people can do that. That's why we end up with 2 rounds of recruitment. Only one candidate has kind of qualified this, but he during the visa application he changed his mind, so that's wasted quite a bit of time, unfortunately. So after quite a few months in February, we talked to VPR office and see we probably don't want to waste more time, and then try to hire a GRA or some other research engineer, which I'm currently supervising 2-3, full-time research engineers to work on the battery research related topic, which we have plenty of opportunity. So currently, I get a PhD and it's coming next week on campus, and then there probably another GRA in 4. But I'm not giving up this hydrogen topic. And there's Weixin. He's probably gonna present it a bit later, and then we are doing something together. So now I was trying to switch the topic to the battery world, which we mostly get funded on, and probably 90% of my work on this field. One topic come to where DO is interesting very recently. That's why I get very busy this few months. Are called a battery for energy storage. They used to push the battery, recharge battery for electric vehicle. And I just noticed a transition. Their SIMs are now fill battery, probably the way eventually going the larger scale energy storage. So sodium ion battery is one of the solutions they are looking for. The beauty of a sodium battery is basically cheap. Lithium battery is nowadays is much cheaper, but it's still expensive. When you buy electric vehicle, yeah, you're gonna still consider the more expensive than an ICE car. But a sodium battery is mostly 40% more cheaper than this lithium battery. And then not just the cost, it actually sodium battery can do fast charging, to charge faster and also running better at a very low temperature, like North Dakota. So, and also currently, because of the huge effort in the Asian, they are trying to boost up the energy density. Some company are now heading are kind of launch the battery, sodium battery, which can with the energy density kind of match with the low end of the lithium ion battery, like lithium iron phosphate battery. So with that, what we are currently doing is we are trying to use the coal again as the feedstock. We are work with our rare earth group, so they kind of extract the rare earths, those metal critical minerals, and then get a upgrade lignite, and then we use that as a cleaner feedstock to making carbon material. Hard carbon is one of the example, and we have funded by Department of Energy quite a few projects about making synthetic graphite for the anode for lithium-ion battery. So this is a new stuff we're trying to do. So I try to emphasize this is a high synergy with rare earth project. Yeah, those guys are exciting too because the economics of the rare earths must rely on the use of the carbon side because the rare earth is expensive, but it's a very low concentration. So you try to find a value on the carbon side of the coal. So this is my kind of a product in the last year about the full proposal. I write 7, 8 full proposal, mostly go to the DOE, kind of NSF, and then just these 2-3 months, we have like a four full proposal. All went to the Mitsubishi ERE, so they are mostly funding the battery work, and yeah, there's one due next week. So, and then there's a very small fund I will try to emphasize a little bit, and that's something new. You eventually we're getting very excited. This is a company a contract. There's a company called Packet Digital in Fargo. Originally, their business is build the battery, to buy battery and for the UAS, we're probably familiar with this pretty well. They are an electronic company, and then they get a lot of defense grant. That's how the business. And now because of national security, and then they are not allowed to just buy the whole battery from China or from other countries. So they just open a facility to actually make the battery, right here, which I can hardly imagine that's gonna happen in 5 years before. Probably a lot of people not even heard about those battery. So now we are very closely collaborate with them. So this is one of the example how we can help this small company to getting growth. So we are trying to work with them very closely on the other project too. And there's the wider paper. Yeah, some of the overlap with the previous full proposal we mentioned. Most of them you're gonna see the white paper. This just a white paper for the ERE. I submitted two actually, and that's you can see the very last two just last night. That's getting very lot of request from the ERE. I think they're pushing this field, for the energy storage. Publication-wise, we're probably not that good on the publication side. We are mostly trying to work with a lot of times working with the company, confidential stuff, and so patent probably is a higher priority than the publication. But we have been working on this quite a few years. So one thing we try to highlight is we have a review paper on the co-derived carbon anode for the lithium battery. So that's kind of summarize what we have been done in the last 7, 8 years now. So the plan is trying to getting into this sodium battery zone. We have a couple people experience in this, but try to get the new guy GRA in this field. And also, yeah, getting more proposal, right? And yeah, there's basically the NSF. Those are kind of annual opportunity for a program. And the ERE or FECM, that's what we mostly get the solicitation. That's come anytime they don't. So basically, That's everything. Thank you.
Hi, everyone. My name is Susan Eliazer, and I'm an assistant professor in the Department of Biomedical Sciences at the School of Medicine. So we got the funding late 2022, and like some of you, we also had problems hiring a new postdoc. So I requested VPR if we can fund one of my existing postdocs, and they said yes. And so I was able to put Dr. Pukana Jayaraman, a postdoc, on this project, and she is going to go over the project that we're working on. And I want to thank VPR for the funding.
Thank you for the introduction, Dr. Eliazer. So I'm Pukana, a postdoc from Dr. Eliazer's lab. So firstly, I would like to thank the postdoctoral seed funding for funding my research. So today I would like to share with you all the research that I'm working on. So the topic today is titled Identifying Mechanisms to Rejuvenate Stem Cell Function in Skeletal Muscle Aging. So as you all know, we all age, we all undergo process of aging. And according to the US Census Bureau, the population of aging in 2019 is 54.1 million, and it is projected to increase to 94.7 million by year 2060. So the severely affected tissue systems in the human body is skeletal muscle. So as we age, there is a loss of muscle tissue, and there is a loss of regenerative potential after an injury. So 40% of a human body comprise of skeletal muscle. And there's this tissue resident stem cell known as satellite cells. They actually important in regenerating new cells. So stem cells here are cells that helps to regenerate to form muscle tissue. So here we identify stem cell as PAC7. So when you see there, that's PAC7 in a muscle tissue. So the function of stem cell is to provide postnatal growth and maturation during development and helps in regeneration of skeletal muscle when there is an injury. Just to let you know, stem cells are in a dormant stage. So when they are in quiescence, in adult stem cells are in a dormant state. It means that there's less activity, there's less metabolic activity. So they will only get activated when there is an injury and they starts to regenerate after that. So we want to find what actually maintains the stem cell in a quiescent state. In our previous lab, in our previous study, we found that there is this protein secreted from muscle fibers that helps maintains the stem cells in a quiescent state. So we come up with a mouse model, which is a tamoxifen inducible gene deletion model. So here we have this model to look at the function of Wind4. So here in this mouse, the Wind4 is deleted in muscle fibers. Upon deletion, they are left for recovery for 2 weeks. And then we analyze the PAC7 cells, which are the stem cells. And we found that in a muscle fiber, which Wind4 is deleted, has increased PAC7 cells or much higher stem cells than the control. Next, we want to look at the stem cell state. So we want to see what happened. What if the stem cells actually changing from quiescence to activated state? So we looked at a marker known as MyOD. So MyOD marker is to be seen less in quiescent cells. When the stem cells get activated, you will see higher of this MyOD marker. So in a muscle fiber where Wnt4 is deleted, we saw increase in percentage of MyOD in stem cells. So this suggests that loss of Wnt4 changes the stem cell state. So in quiescent state, MyOD or activation is low. When the stem cells activated, its MyOD marker increased. So here we want to see the function of stem cell. So therefore, with the same model, we have an injury here. So upon injury in one of the leg of the mice, we looked at the fiber size. So we can determine the regeneration in this model. So when you see here, in muscle fiber where Wnt4 is deleted, there is a better regeneration because the fiber size is much larger than the control, which is on the left. Since aging is associated with impairment of regeneration, we want to study an aged mouse model. So we have the same model where which the mice is about 20 to 22 months old. And we injure this mice. So what we saw here is that the fiber size are bigger in muscle fiber where Wnt4 is deleted compared to the control group. So this shows that stem cells are better at regeneration. So we wanted to find out what is the mechanism behind this? What is causing this regeneration? So there is this pathway. So what we did is we did an immunofluorescence staining of the signaling pathways, and we get to know that Wnt4 regulates the ROE-ROCK signaling pathway. So we want to know how Wnt and ROE helps in regulating the stem cell. So since ROE is a downregulation of Wnt4, now we perform the same experiment to see if it is a downstream effector of Wnt4 that helps in the pathway. So here we have a mouse where we delete the ROE only in stem cells. So we found that the stem cells increase in ROE deleted stem cells compared to the control. So here we perform to see the activation of the stem cells. What happens if ROE is deleted in stem cell now, to see if ROE is really a downregulation of Wnt4. After deleting ROE, we looked at the percentage of MyOD, which is an activated marker, is to be seen increase compared to control. So this shows when we delete ROE in stem cell, it shows a similar phenotype when we delete Wnt4 in muscle fiber. And this agrees with Wnt4 actually, ROE is actually a downstream of Wnt4. So we wanted to, now that we know that ROE we know that Wnt4 regulates ROE in the Wnt4 regulation pathway. So we wanted to look at the stem cell identity. So we wanted to study DNA methylation. So what is DNA methylation? DNA methylation is a chemical modification that actually adds a chemical tag known as methyl group that added to the nucleotide of DNA. So this doesn't make any changes to the DNA sequence, but it affects by turning on and off some of the genes. So here when we talk about DNA methylation, we are studying the enzymes that add the, uh, the, the metal mark to the DNA sequence, which are DNMT3A, 3B, and there is another uh, um, enzyme known as TET1, 2, and 3, which removes the metal mark in the process of DNA methylation. So here we are looking at the DNA methylation landscape. So what we did is that we sent a sample of genomic DNA from a mouse, a control mouse, and ROA deleted stem cells, which ROA is deleted for EM sequencing. So when we did the sequencing, we saw some of the genes are hypo and hypermethylated here. So here we found that ROA actually maintains the DNA methylation landscape in quiescent cells because there are some difference in differential expression in the ROA deleted stem cells. So when I talk about the enzymes that adds the metal mark to the DNA, so this is the one. So there are 3 different enzymes that plays a role in maintaining the stem cell in quiescence. So we wanted to look at DNMT3A. So we looked at the markers that add the metal group, that is DNMT1, DNMT3A, and DNMT3B. So here we saw that DNMT3A is expressed much lower compared to the control group, comparing to the stem cells, which ROA is deleted. And in stem cells where ROA is deleted, for DNMT3B, it is increased. And this is TT1, a protein expression that we see, which removes the metal mark from the DNA. So among these 3 types of TT we have here, only one, which is TT1, to be seen increased when ROA is deleted in stem cells. So to conclude my work, so we know that Wnt and ROA pathway actually maintains the quiescence in stem cell. Okay, it's doing that by regulating this DNMT3A. So we further want to look at on how ROA regulates DNMT3A by knocking down DNMT3A. So we feel that that is our future experiment. So we feel that DNMT3A could be a potential target in aging and disease. So last year, we applied for 2 different grants. One of it is Evolution Foundation, and the second one is NIH R01 grant, which the second one, R01 grant, we plan to resubmit again this October. Hope we get this grant to support our research. And this is our plan. We are planning to submit 2 articles this year. We hope we can submit these articles by end of this year. And I would like to thank the VPR for postdoctoral seed funding, and also my PI, Dr. Eliazer, my co-workers, Atrey, Christian, and also I would like to thank those imaging the course who actually helped in supporting my research. Thank you so much for listening.
We have one more. And so after this one, we'll do questions for all them. We'll take a quick break and then do the newest cohort.
Yes, thank you. Yes, please. Um, Hi, everyone. Right, I'm Utkarsh. I'm the postdoc at Department of Psychology, and I'm working with Dr. Dmitri Poltavski and Dr. Thomas Petros. So I'm pretty new. I only started from the 1st of January of this year, and it's been only 4 months of me doing work here. But I think there are lots of progress made around that. So a small snippet of what I was supposed to do from a job description is the fact that I was supposed to help Dr. Poltavski and Dr. Petros with the existing research, as well as help with grant applications, and also come up with individual and collaborative research projects, not just the existing one, but the individual ones that I would be taking lead on as well. And apart from that, it being a collaborative approach, like I'm supposed to come up with grant application, not just within the UND or NSF, but with potential links of industry as well, if there's a potential chance of that. So I'm going to talk about the theoretical focus for my postdoc position here in the coming slides. And I'll talk about the overview of what has been done. I'll walk you through what I've done so far. So in terms of processing the existing data, and then talking about the new tools that we've got and how we'll be using that in our research. And then a couple of IRB applications that got approved, and then we plan to work on it during the summer or after. The grant application process that I've worked on, and other industrial outreach that I've worked on as well. And then I'll quickly give you a snippet of what's next from there after these 4 months of work here. Right, so Dr. Petros and Dr. Poltavski, they have been extensively working on working memory and mental workload capacity. So what I mean by working memory is, say, for example, we are having a conversation right now. We are thinking about me speaking right now, standing in front of you, someone having a coffee, or someone making some note. The executive function is happening there. You store certain information, and then you use and manipulate it, and then essentially have fun with it. That is working memory. And the mental workload capacity is associated with working memory as to the capacity to which we can actually play with that information, the stored information, and manipulate it. But as a postdoc, what was I meant to do? What would I bring to the research here at UND? So during my PhD back in the UK, I worked quite a lot on memory, as in autobiographical memory, and bodily self-consciousness. And both of these cognitive faculties integrate quite a lot of multisensory integration. So contrary to what usually is thought of having a brain region associated with some sort of cognitive function, recent research and literature focuses quite a lot on multisensory integration, because when we talk about bodily self-consciousness, it's essentially about your fundamental sense of being, your fundamental sense of consciousness, where you relate your existence within a body. And while I look from a first-person perspective, or have a sense of ownership, and then, or like self-location, where do I look at myself or like feel to exist in this experiential space? So these all are contributed to senses of perception, like whether it's visual, auditory, whether it's vestibular or other sort of senses. And all of these exteroceptive, that is from the outside, or interoceptive senses from the inside, contributes towards the sense of consciousness. And then from working memory, when this body impacts this memory manipulation, it then leads to how some of these functions, like riding a bike or driving a car, becomes more automatized. So there is this automatic functionality that is being driven towards, and that is the procedural memory. And what we want to do throughout my position here and forwards as well is to look at the interaction between these cognitive faculties. So, like I said, what I've done so far, while doing my postdoc position, I have already helped Dr. Petros and Dr. Poltavski with certain pre-existing data, raw data, managed it. And then there are a couple of equipments, hardware and software, both, that we worked with, and we are going to apply that in our upcoming research. Like I mentioned, we have 2 IRB applications sorted out, and we want to start the research around it. Grant application, something that was led by Dr. Petros initially, and we're still working on that for its resubmission through Spencer Foundation. But I'm taking lead on NSF Cognitive Neuroscience grant, which is a 4-year plan that I propose, which I'll talk about in a bit. And then industrial outreach, like a couple of other postdocs here who have participated in ICORPS. I participated in ICORPS as well with Dr. Poltavski, but there was another additional collaborative approach in which, like, we had Dr. David Bibadov, an ophthalmologist, collaborate with us, and we were working on this medical screening device based on Pulfrich phenomenon, and then doing the customer discovery around that. And now we are in process of applying for NSF grant through SBIR, that is Small Business Innovation Research Grant mechanism, or SCTR as well, which is Small Business Technology Transfer Grant. And then, like, yeah, like I am very entrepreneurial. Like back in the UK when I was doing my PhD, I was still working on a startup which was neuroscience focused. I resolved it in the UK since I'm here in the US now. And I pitched my neuroscience startup here at the Chambers Shark Tank, which happened just on the 30th last month, where I bagged the second prize. So that was fun. But what I could provide with that is essentially another link with industry, and we could have more SBI or SCTR applications that we could work on. Right, so the processing of data, which includes quite a lot of R coding and then essentially managing the raw data collected. We did quite a lot of raw data management for electrophysiological data from EEG. So essentially all the brainwave activities that is happening, and then essentially making sense of it while we have certain cognitive functions in play. And then Corner CPT was one of the tasks in one of the vision study, which essentially looks at the attention of participant. And then we were looking at the electrophysiological correlates during the attention and mental workload capacity during that research. It's still under process of analysis, so I don't have any results as of yet. And since I'm pretty new here, so I guess like it will have to wait. Then in the meantime, we've worked on the experimental tools. So this one right here, that's BioPack system. So if you remember the cognitive faculties that I talked about, and I introduced the idea of multisensory integration. So the whole idea is that simultaneously so many sensory inputs are being flown in our body that we have to kind of get everything, the bigger picture together. So BioPack is a system in which we have electrodermal activity. We could link it with EEG. We could have also heart rate variability and other like respiratory system being measured as well. So quite a lot of facilities are brought in together, and you could use that while experimenting, assessing the multisensory measures. And then the software here, again, the multisensory aspect of things is, so this one is a MAT-B. So this is a NASA software that Dr. Petros acquired. And then we've been working on this because all these sections are a different task that a person has to perform simultaneously. And this is Sinwin, another multitask attribute cognitive task. So here again, these little blocks are different tasks that needs to perform together. So again, the focus on multisensory. So essentially my entire postdoc is being sort of focused on the multisensory processing within the human brain. So this was about the new experimental tools. So hardware and software, everything together. We're having fun with it. Then the IRB application, again, very electrophysiological. We have quite a lot of neuroimaging tool and neuromodulation tools. So we have EEG as one of our prime neuroimaging tool. But apart from that, we have TDCS in our lab as well for neuromodulation, in which we passed some sort of electrical current in our brain to temporarily manipulate the cognitive function. And along with that, we have some other tools like FNIRS, which is like a small brother of fMRI. So the blood oxygen level is kind of calibrated through that. So, but we are not doing that here. So these two projects were approved, and we are going to start recruiting participants for it pretty soon. We're talking about the grant application. So Dr. Petros had already started this application for Spencer Foundation, which is essentially a project that looks at assessing the functionality of people while in a lockdown. So essentially the distance learning while Zoom. So what kind of attentional factors are in play? How can we enhance distance learning? Because a lot of people now just jump in Zoom rather than coming to the classes, and like universities all around the globe as well, they are kind of adapted towards this hybrid nature of teaching. So of course, like we have to work our way to understand what is exactly happening. And that is all about Spencer Foundation. We got some reviews and then we are resubmitting it right now. Now, my little baby is the NSF Cognero grant. So this is a 4-year plan in which we have 2 phases. Phase 1 is where we will be investigating the link between working memory and procedural memory, and investigating the behavioral and neurophysiological indices for it. And then in phase 2, the next 2 years is when we will be applying what we have found in real-world applications. So essentially, how we could enhance procedural memory or working memory in population of aviation, for example, since it's a UND specialty. But not just aviation, but healthy individuals and expansion in clinical population as well with memory impairment. But it would not be just simple experiments with potential neuromodulation of the indices that we have found, or like the brain areas or the connectivity that we have found to be playing a role in working memory and procedural memory. But we're also looking at brain-computer interface application in the last couple of questions in this grant. So we have around 15 to 16 questions structured for this grant. And yeah, so brain-computer interface application, live data collection, and manipulation of cognitive abilities is what we are targeting by the end of this grant. And so we have a deadline of 15th of August to submit this grant. We are now working on that 1-page summary to send it to the program director of NSF Cognero grant, and hoping to hear back from them as soon as possible and then finalize the grant and submit it by 15th of August. But we are looking pretty solid with that because we have everything sorted out now. I'm just waiting on a few of the quotations for what I need to get the grant money requested for. Well, so apart from the grant application and everything, like I mentioned, I am pretty entrepreneurial as well. So this was a certificate for ICOR program, which is again sponsored by NSF. And because NSF strongly supports the link between academia and industry, and they have been helping with the SBIR and STTR mechanism of funding quite a lot. And that is what we got to know about it. And so it was not just the project. So there are so many projects going on in my postdoc right now. So we have got this whole working memory, mental workload capacity, medical device funding application. And then this is the picture from 30th, the thing that I mentioned about the Cambridge Shark Tank, Chamber Shark Tank. And essentially, like I got so many good leads there. Like I was introduced to Mr. Phil Giese, CEO of Edgewood. And then I had a really nice chat with Mr. Landon Ball, who has been like involved in entrepreneurial stuff here quite a lot. I got in touch with Mrs. Sean Riley, who has been the Chief Information Officer of North Dakota for a very long time. And he was very interested with my research and the link of it with industry as well. I'm already in the process of registering my startup here. So I've processed it and I'm applying for the LLC there for my venture studio. So the idea is to build a venture studio in which we'll be working towards memory and learning enhancement and supporting other startups within it. So I'm looking for, lots of collaborations, lots of potential investments, grant applications, and really working towards the enhancement of our human function, because it has not just got enhancement function for normal healthy individuals, but so much more for people with impairments with memory and learning or other things. So definitely I'm a big supporter of translation of academia to industry, and I would do as much as I can in my capacity to fulfill that. So yeah. So what's next? Like I said, so what I've gone through, collection of data, and then applying them to the research, multiple grant applications, all the other grant opportunities. And of course, in the meantime, support the current students with research assistant position. With the grant application, we could have new postdocs as well. But of course, now that I'm thinking about the industrial link, of course, there's a lot of development, not just for UND, but in North Dakota in general, that we could bring in more economic development via what we are trying to achieve here. So that's about me. Thank you so much. As I said, please do get in touch if you've got ideas or collaboration opportunities. I'll be very welcome. I'd be really happy for that. So thank you.
Thanks. We'll have questions for any of the groups now, and I'll hand you this microphone. It helps with people listening on Zoom.
Well, hello. I have 3 questions. First question is for Karsh. I'm pronouncing it right? I'm sorry. So my name is Tarek, and I actually want to ask you, did you consider collaborating with biomedical engineering or SEEK, School of Engineering Computer Science, regarding your research project, since you're working with some sort of voltage and current for the brain?
Yes, so throughout my conversations, I've already had an introduction with Dr. Kuya, and I'm hoping to have a conversation with him this Friday. And yeah, like I'm really looking forward for that. But like if you're interested to have further
As well. So
okay.
that's perfect. I was just wondering if that's some sort of a thing that, yeah, okay, sounds good. Awesome.
The industrial link, yes.
Awesome. Thank you so much. And the other question is for Paul and Trijesh. Mike, hi, welcome. Welcome aboard. So, Paul, you said that you want to have, you're looking for students to work on the projects you're working on. So we have in SEEKS electrical engineers with biomedical focus and as well with aerospace focus. So we have a lot of students that are going to be interested in working. If you have any opportunities to hire students for aerospace or even work on research as students for research groups or senior design project or stuff like that. Also, we have faculty members working on drones and path planning, collision avoidance for drones as well. So be a good chance if we can have some sort of collaboration on that.
I think that would be fantastic. I'd love to.
Sometime. Thank you. And for Pantea Rana, just last question. Regarding the early promising detection for the source, do you have any kind of expectation how early is it going to be compared to the other research work?
Yes. So the SEM device is providing 5 days earlier on just one biomarker. So the device we're developing is going to have different biomarkers. Apart from moisture, we're going after oxygenation, which gives us information about blood going to the skin. We're going after collagen, which is a mechanical properties of the skin. And we're erythema, yeah, pigmentation is something, redness on the skin, quantifying that. So one of the challenges that exists is with darker skin tones, the redness, the skin visual assessment has challenges there, and that's why the number of people, the number of pressure injuries occurring for darker skin tone is twice often than lighter skin tone. So pigmentation is another biomarker we're going after. Combination of this will give us a higher accuracy than SEM, and therefore we don't know it now. This is what we're going to try with clinical studies that we are planning by submitting the IRB. We don't know it now, but it's promising.
I can tell it's promising since you're using multiple of techniques. Actually, that's going to be opening the opportunity of early detection than the others, and that's what I wanted to hear. That's coming up. Thank you so much. Appreciate it.
Okay, our last speaker was it Utkarsh? Okay, super. So, okay, I love that you're going through the I-Core, the whole 9 yards. When you did your Shark Tank, what are you going to sell?
Oh, so
What's the product that's going to come out of this?
it's a venture studio. So because I'm working in memory, and I can have my virtual reality and augmented reality coupled with neuromodulation research as one of the portfolio startups. But apart from that, like those biomedical engineering startups that could actually make a compatible, say, for example, VR with a neuromodulation device together, or some other form of startups as well. Because I was thinking about this, that one could actually have virtual avatars to enhance memory and functionality based on my research, which I could talk in detail. But there are multiple portfolio startups that I want to support because I can't do everything together, and hence the venture studio business model towards it. So the main umbrella goal of the venture studio, so it's called the Neurogather Ventures LLC, and its idea is to enhance memory and learning through different portfolio startups.
Got it. Okay, thank you. That's awesome. Keep up the good work. Yeah, very good. Okay, Pukana. So I know in human beings we start losing muscle mass at around 30, 35 years old, something like that. Do you see a loss of muscle mass in all mammals at some corresponding point in the life in their life stage? right? So in other words, do mice in nature lose muscle mass at the same time in the average lifespan that humans start to lose muscle mass?
Yeah, so the mice also lose muscle mass, but then they increase fat in response to loss of
I
muscle.
resemble that remark. So,
So yeah, the mice are pretty big.
okay.
Yeah,
Okay.
but they do lose muscle mass.
And does that seem to be the case in all mammals where this have been studied? This is just sort of something that happens as all mammals age?
I would say so. Um, I I don't know in detail about all mammals in general, but human beings for sure
Yeah.
do lose muscle mass, right? So
Okay. Okay. Thank you. Xiaodong, okay, I have a question about yours. And so you talked a good bit about sodium ion batteries, right?
Yeah.
So my question is why don't we see sodium ion batteries here or in my car or whatever it is?
Either way-
What are the limitations to sodium ion batteries?
Yeah, either will come. I think in China now you're gonna see sodium battery powered vehicle. There's some historical reason. Actually, sodium battery were most being researched at the same time as the lithium and lithium battery. And just because people did not find a very good anode materials for quite a long time, so eventually, uh, the scientist who won the Nobel Prize a few years ago, they figure out graphite is the ideal anode for that's by Sony and make it the battery work. So the lithium become the front runner, and apparently the theoretical energy density is higher than. So people just discard sodium, from there. And but recently you see the lithium because the price is kind of skyrocketed for a few years. Now people are getting back, and then now the knowledge is getting better. So people are thinking, yeah, sodium battery definitely gonna be play a bigger role. Even the energy density is lower, but there's some good side, like what I mentioned. Cost is one thing. Low temperature performance is another thing. You probably heard in winter last year, a lot of Tesla, vehicle done the start. Yeah. And the charging is another issue. People still don't like EV because take maybe 20, 30 minutes fast, right, to charge it. And you don't want to charge a whole night to get it running. So, but those things, sodium battery can easily solve, So the only thing is, the energy density lower, so might be not like a passenger car, like but it's for like a short distance vehicle, those are or especially motorcycle. And I think the major target is energy storage because it's cheap. And then the cycling life is much longer, probably can be very long. So overall, the ownership cost is very low. That's good for, and I think that's why Department of Energy now is getting interested in this. I did not see any, major funding opportunity about a sodium battery just a few years ago. There's nothing. They don't care about this. They all pursued for the electric vehicle. This sodium, this lithium battery. So that's
Excellent. That's really, so it's kind of a historical artifact in some ways. Yeah.
kind of.
Yeah. Cool. Thank you. Thank you. Let me see. Okay, Rana, you were talking about going through the I-Core process and maybe SBIR. So assuming your clinical trials go well, assuming all of this happens, what's your most optimistic timeline for a device that does this being on the market?
From the previous experience I saw with the SEM and how the research started, it's been taking, I would say, 4 to 7 years. So yeah, it's not less than 4 years. I think so.
Okay.
Yeah.
Okay, that's good. No, it's fascinating what you're doing. And I love this. I love the skin pigmentation that you're trying to control for that as well. But I was just curious as to how long it takes. So
Yeah,
that's
yeah,
awesome.
so we have this FDA approval, or I think it's a process. So I think we're looking for almost 4 years. Yeah,
Right. And the FDA approval is probably quite lengthy.
I'm not sure. And I can help with that.
So the good thing here is that we're working with a company, and that brings its motivation to it. So, and on the other hand, we might even have an addition to it, and a patent might come from the work that we're doing with the company. But yeah, it's a lengthy process.
Right.
I don't say so. With one of the devices that came to BME, that it started from scratch. I think it's been 5 years they're working on this project, and they're getting closer to commercializing that device. We're not there yet, even for that.
Thank you. Thank you. And then finally, Paul and Mike. Mike, welcome. Okay, first question is, you mentioned some funding sources. I didn't hear Department of Homeland Security. Is that an option? I mean, this counter US seems like a big thing
Yeah,
for
we're
DHS.
Doing work in that area, but not particularly with Mike yet.
Right.
On some projects.
Okay, excellent. And the second is a request when you mitigate, which means blow up or otherwise bring down, right? When you do some of this counter UAS stuff and you're doing it either inside or in a controlled outside environment, I want an invite. Okay. All right, beautiful. You know where to find me. Thanks, everybody.
Questions for this cohort? All right, why don't we take a few minutes break, and then we'll come back and hear the second cohort. Thank you. There's snacks in this other room. Great. Yeah. All right, everyone. Hope you had a chance to get a snack, and we'll start back up again. So the order will be, we'll have David. You'll be next after. Then Ali, you'll be third, and Manu, you'll be that. And then after those four, we'll have another session of questions, and then VPR will give closing remarks, and we'll call it a day. Thank you, everyone.
Yeah, good afternoon, everyone. My name is Weixin Huang from Chemistry Department. Yeah, my name is difficult for people to pronounce, and then typically in my courses, I will tell my students I'm from chemistry. It's very important to find a good way for synthesis. So my name is Weixin, so good way for synthesis. So easy for people to remember. So thank you for giving me the opportunity to introduce our work. And then I'm the PI, and Dr. Zhao is the co-PI for this project. And then our proposed title is develop a novel catalyst for ammonia synthesis. So when we talk about ammonia synthesis, so we need to talk about the nitrogen cycle. So people know about the carbon cycle a lot, but nitrogen cycle is maybe not very familiar with. The nitrogen cycle is we convert the reactive nitrogen source to the ammonia. And then ammonia is very important for the biological energy, right? So it's very essential for making fertilizers. And then it's important for our daily life. And then if you check the data in the past, so actually the ammonia's productions strongly correlates with the human population in the past centuries. So right now, the methods for making for ammonia productions strongly relies on an energy-intensive process. And then we call it a Haber-Bosch process. And it operates at a very high pressure, 200 and 400 bar. So we know the 1 bar is atmospheric pressure, so which means 200 and 400 times higher than atmospheric pressure. And also at a high temperature, 400 to 600 degrees Celsius. So typically, this kind of a process consumes a huge amount of energy. So approximately 2% of the global energy in a year. So it's a huge amount. And also emits like 500 million tons of CO2. So this kind of process result in energy and environmental concerns for us. So to find alternative solutions, so we would like to develop a catalyst which we can mimic the structure of nitrogenates, and then for the ammonia synthesis. And then we have questions: what's the structure of nitrogenate? So in nature, nitrogenase is an enzyme capable of fixing nitrogen at ambient conditions. So not at high temperature, not at high pressure. So that's great. And then its active structure in this kind of enzymes has been identified as a isolated iron and molybdenum single atom structure. So it's this kind of a chemistry structure capable of fixing the nitrogen. And then the ideal case, if we can develop a catalyst, we have the similar structure, make this kind of structure, and also have the same function. And then we would like to do that. So how can we do that? And then with the development of nanotechnology, so numbers of nanomaterials with this kind of enzyme-like characteristics has been reported to emulate the enzyme's functions. And then this kind of nanomaterials is called nanoenzymes. And actually, this kind of materials, Dr. Zhao's research group have developed a lot of this kind of nanoenzymes. And then we would like to use, inspired by this nanoenzyme idea, so we like to develop the catalyst, have the atomically dispersed iron and molybdenum's single atom size. And then we can, for the ammonia synthesis. And this kind of design catalyst, and then with the features, firstly is controllable bounding environments for this kind of single atom size. And second is unsaturated coordination configurations. And then the third is a tunable ratio of these two single size, iron and molybdenum. And this kind of special features can make this catalyst potentially have the function for ammonia synthesis. And then right now for this project, we are in the middle of the hiring process. And then as long as we can have our postdoc on board, and then we can work on this project. And then Our target fundings will be mainly focused on NSF and DOE. They provide a lot of grant opportunities. For example, NSF have critical aspects for sustainability, and then we can, as long as we have preliminary data, and then we will submit applications in this kind of funding opportunities. And that's it for our projects. Thank you so much.
Okay, I'm David Delene from Atmospheric Science and Aerospace College. So our project is entitled Autonomous Systems and Data Science to Support Airborne Atmospheric Observations for Research Project related to Cloud Physics, Stratospheric Operations, and National Security. So I got all the key points of what we're doing at UND in my title. But we are just starting. So as first cohorts had expressed very difficult to hire people. So I actually started before I even wrote the proposal, trying to hire people. I talked to two people, talked to a bunch of colleagues to try to get people lined up. And I had two people interested in the project before I even wrote the proposal, or else I wouldn't have written the proposal because I knew how hard it was. So I know you guys are all struggling with this, and it's just difficult. So anyway, I'm still working on getting my postdoc hired. Been working on this since we got the award. So that's one task I've been doing. The second task on this project is, while we're focused on doing proposals, we're going to do some preliminary data observations this summer with the aircraft. So I said airborne measurements. So we have historically, this is building upon historical stuff we have done in atmospheric science, which is observations from aircraft and aviation. So we're going to do some cutting edge measurements with a new instrument that's never been flown before this summer. So I've been planning for that. Also, we've been planning for proposals. So I don't know if I'll be able to keep up with Paul in aviation with a proposal a month. I'm thinking I'm going to target a proposal every 2 months. So we have some ideas, and actually I'm going to miss a call tomorrow. There's a notice of intent that if I would have had my postdoc working like Paul, he would have wrote this notice of intent this week, but not doing that. So one proposal mix, so cross that number 0 out. However, NASA has a call that just came out. We got a meeting, town hall meeting about that for software development based on existing proposals. People that have existing proposals can write. So that's one proposal to NASA. We're going to focus on Department of Defense, Navy. They're interested in cirrus clouds. I can tell you the reason why that is. But we want to do some proposals with them, 1 or 2, Navy, Air Force. I got another proposal idea. Recycling a previous proposal to the National Science Foundation about how do we do tracing measurements in the clouds so that we know what we're measuring and how they're affecting the clouds. I got like a lot of people said, I'm collaborating with a company. They own the aircraft. They own a lot of infrastructure. So I got a meeting next week with them. We're thinking about doing an FAA proposal. We're waiting for aviation weather. We've been talking about that. And then that's 3 proposals that we're having. And the 4th one is with the preliminary data, we also want to look at serious and climate change type of stuff. So then what we're doing, we haven't done anything really much yet. So that's the summary.
All right, Alshami from Chemical Engineering. I'd like to actually, first of all, thank you for the VPR office. Thank you for the award. We're very happy. Also, I don't want to make you feel jealous, but we were not as we did not struggle as much as you guys in finding a postdoc. Our postdoc just happened to fill in our hands right away. So Dr. Pantea joined us about 4.5 months ago, even though we were still in the planning of the actually idea of the proposal, but we were also had some ongoing projects. So Ban comes to us from Jordan, the University of Jordan. Prior to her opportunity with us here, she had done her postdoc about 2.5 years at University Christian Albert University in Kiel, Germany. Yes. And I think your PhD from University of Jordan as well. Yes. She has been very active, I would say, and we're fortunate she can tell a little bit more, but I think I got her started before even arriving here. And she has been working, I think, what, 2-3 proposals now? Proposals? Yeah, two successful. We have a few in the pipeline. We have one with NASA. We have one with NSF, and we're working on a few other ones, in collaboration. So it's a godsend for us that Dr. Ban joined us because we're relatively large team. I'll tell a little bit more about that. So, Ban. Yep.
Ali, hello all. Thank you for having us here today. Actually, I'm involved in six research projects in Dr. Alshami's research group. So I'm involved partially. I will just mention for those projects. I want actually to do a presentation today because we already have data. We already have promising results for some of those projects. For the first project, we have one PhD student who works on machine learning as a tool for material discovery, especially for water technologies. And this PhD student works on studying the performance of RO membranes, and his work resulted in prediction of promising polymer materials for reverse osmosis membranes applications. So my role in this project will be in assisting in the experimental part in order to evaluate the results obtained from the machine learning studies. In the second research project, we are working on polyimide membranes for gas separation. And so far, we successfully synthesized the polyimides and we already characterized them. We found out that the average molecular weights of the material we got are better than the commercially obtained ones, and they have better, let's say, properties than the ones we bought from the market. In the third research project, we're working on hydrogels, scaffolds for biomedical applications. And in this project, I'm working on assisting actually in the synthesis of those hydrogels. And at the same time, I'm working on studying and investigating the influence of the physical cross-linkage of those materials on the morphology and thermal properties of the hydrogels. The fourth research project, we work on the development of antiscalant materials, more environmentally friendly antiscalant materials based on bio-based building blocks, polymers from bio-based building blocks. And in the fifth research project, actually in the sixth research project, I'm working almost individually on the synthesis of bio-based materials from building blocks derived from soybean. Actually, we are working on gallic acid. It's a unit derived from soybean byproducts, and we are synthesizing a material for coating applications. Thank you.
So, so this proposal was with me. My name is Manu, and Benjamin Roche, who's in SMHS. So Dr. Nuti is a postdoc with me, who's a recent postdoc, and he kind of came up with combined elements of our lab's work and Benjamin's work to come up with a new research direction. And so our proposal was based on this. And once we got it, and thank you so much, we requested an exception from VPR Snyder and Dr. Berg here to, given that it's a new research direction, to allow us to support Dr. Nuti. And so he is sort of already working on this project, and I'll let him tell you about it.
Thank you, Manu. thank you, the VPR office, for this opportunity to extend my postdoc. I've been working with Manu on his career grant, so where he works on enhancer interference. So our cells are made up of 3 billion base pairs, of which only 1% of this is coding genes. We have remaining everything is a non-coding gene. So we work on all those non-coding genes, like how they decide what the stem cell becomes. So in our body, we have about 30 trillion cells, of which 90% of them are like blood cells. And all these blood cells are again have a very short lifespan, which means that some cells live only for a day or two, whereas some live for. How does what Manu was working before was how does this non-coding DNA decide how the stem cells become red blood cells or white, etc. So what I have been fascinated from my previous earliest work was that these blood cells have to divide a lot. So unless until you divide, because they have a short lifespan, and you have to have like divide and also differentiate. Differentiate meaning stem cells has to become white blood cell or a red blood cell or a platelet. So how does so this question of how do cells decide when to exit the cell cycle and then become came up with. And what Manu does is he's a physicist, so he does a lot of modeling and gene modeling, network modelings. So we have a cell fate process, which has one type of gene regulatory network. And now we have a cell cycle process that has another gene regulatory network. So what we are proposing is to see how cell fate, which is decided by some unique transcription factors, like for example, we have around 400 different cell types in our body. So each cell type is decided by some transcription factors which will make expression or not. So how do these 400 different cell types, which are going into becoming cell, how do they decide when to stop division? So if you don't stop division, then you can keep on dividing and then renewing yourself, basically the process of the stem cell. So what we proposed was that we will take this gene expression data of dividing cells versus exited cells. And we have a small model system where one stem cell will become either a white blood cell or a cell in our lab. So we have collected data, time series data for about 7 days. He does a lot of modeling, and what I'm doing is I'm generating good data so that we can have some predictive models based on machine learning. So I hope to learn some machine learning as well while I produce all this data.
Thank you so much for those. Time for questions. Who would like to ask a question of one of the teams? Hand you the mic. Yeah, thanks. Start us off.
Yeah, I don't necessarily have a question. I do look forward to those of you who just talk to having either you report out or the postdocs that you do ultimately hire report out next year. I'm really looking forward to that. This is just a really fabulous event, and you get to see all the big brains talking to one another. What I really like about stuff like this, though, is then I see people introducing themselves to one another, and you start to get these cross-disciplinary opportunities that might not happen otherwise, or would be more challenging otherwise. So I think this is, even though, you may sit here and say, I'm not really quite sure what that person's talking about, but something out of there often resonates with the world that you understand, and then you can start to make those links. So this has just been a very powerful and fascinating afternoon with you, and I look forward to seeing you all around and learning more about what you all do. Thanks.
All right, thank you, everyone. We'll just call it an end here, and really appreciate you sharing your information with us, and we look forward to hearing more about it. We'll see you next year. Thank you.